Clusters and subclusters are efficient ways to categorize phages based on their genomes. Through our studies, including a well-written paper by Dr. Jacob Serra, we learned that a phage is placed into a particular cluster if 35% or more of its genes are shared with the other members in that cluster. Clusters are further divided into subclusters, which allow for more specific grouping of phages based on the distinguishing genes they share. We wanted to apply this principle to the EA cluster and discover what separates subclusters EA5 through EA11. Our research determined that their differences in genome length, ORF count, and ORFAMs separated them into subclusters, and that synteny was conserved within subclusters but not across the cluster as a whole. Famirator allowed us to determine the function of individual genes and observe genome length, the prevalence of ORFAMs, and differences in genes across subclusters. Tables were created for a horizontal comparison of the genes within subclusters, and Morpheus allowed us to determine a quantifiable similarity score through percent identities. Maps showed a connection between all the subclusters by using phylogenetic trees. Those trees showed that subclusters EA5 through EA11 belong to the EA cluster despite their differences. NCBI was helpful in distinguishing more differences between the genes and confirming what Famirator showed.
Famirator showed that different subclusters end on different genes. For example, UE ends on gene 64 while Nebulus ends on gene 57. Phages in the same subclusters tend to end around the same gene number, but vary when compared across subclusters. ORFAMs found in all of the subclusters and are represented by the white boxes. Some genomes in different subclusters have ORFAMs in similar locations, but for the most part they are strictly unique to the phage itself. Teresita has 12 ORFAMs, making it the phage with the highest number of them. Our original research showed that all genomes in subclusters EA5 through EA11 lacked a capsid maturation protease gene, but more recent findings have proven otherwise. Phages Schubert, Cheeto1, Chicken King, and Gacio all contain a capsid maturation protease gene at about the same region.
We used Morpheus to create heat maps that show how phages are clustered together. Darker shades of red represent a higher degree of similarity between genomes, and darker shades of blue represent a higher difference between them. This heat map shows each subcluster grouped according to their respective phages. In the top left corner, we observe the EA10 subcluster, which shows the newly discovered novel phage Mandalorian. Based on the heat map, we can see that Mandalorian shares a 100% identity with Quartz and Nuki. UE has slight variations in its genome, meaning it does not have 100% identity with the other phages. Heat maps can also be used to compare individual genes within different phages to highlight the diversity of genes across subclusters. In this heat map, Morpheus clustered the phages together based on similarities between genes instead of based on subclusters. This shows that there is much diversity within subclusters for gene 15. However, even though the other phages were not ordered together based on their subcluster, EA10 phages were. In the bottom right corner, we can see that EA10 phages were still clustered together for gene 15. Interestingly, William Strong, a member of subcluster EA6, was also clustered with EA10, showing similarities between gene 15 of William Strong and the EA10 subcluster. To prove the theory that there is diversity within subclusters for gene 15, we manually ordered the phages based on subclusters instead of similarities between genes. A very different heat map is produced. We can see that there are not as many clustered red boxes when the phages are grouped into subclusters. This proves that gene 15 is very different within subclusters. One of the things that we learned from these two heat maps is that we cannot define a cluster by looking at an individual gene alone. Based on this heat map, we would not be able to accurately define subclusters. We have to look at the genome in entirety in order to properly define subclusters.
The most important takeaways from our research are 1. The genomes in EA10 have the most similar subcluster within the EA cluster. 2. The lengths of each genomes vary. 3. Individual phages, not subclusters, vary with ORFAMs. And 4. Some of the genomes of EA, in fact, do contain a capsid maturation protease gene, contrary to the findings of research in Dr. Jacob Serra's paper. After thorough analysis of subclusters EA5 through EA11, we have discovered that synteny is conserved within the subclusters, but not between them. This is due to the presence or absence of different hypothetical and functional proteins between the subclusters. Therefore, these EA clusters are formed according to their similarities in gene order. A special thanks to these individuals and to the BYU Phage Hunter class of 2021, and I'd also like to give a special thanks to C Phages, the Howard Hughes Medical Institute, BYU, and BYU's MM Bio Department. Thank you.